Bismillahirrahmanirrahim. Now this is a specimen paper for actually 0610, but it has topics which are very similar to 5090. So doing this paper will help you in both the different syllabuses. If you're taking the 0610, it will help you in that and also help you in the 5090. Question number 1. Catalase is an enzyme found in plant and animal cells. It catalyzes the breakdown of hydrogen peroxide to form water and oxygen. Now, catalase is found in all plant and animal cells. So that is the thing that you've got to remember. And what does it do? It produces water and oxygen. So oxygen is a gas, water is a liquid. The oxygen produced forms a foam. You can measure the height of the foam to determine catalase activity. So if we look at it, you've got to visualize that you're actually doing this in the lab. A student investigated catalase activity in cooked and uncooked potato tissue. So we have decided that we're going to take cooked and uncooked potato tissue. The student, step 1, cut 2 potato cylinders so that they were identical in shape and size, just like when you cut chips. Put 5 centimeter hydrogen peroxide in a test tube labeled cooked potato. So you took a test tube and you labeled it. So put 5 centimeter cube of 3% hydrogen peroxide. So we've got 3% hydrogen peroxide in this in a test tube labeled cooked potato. So this is the cooked one and this is the uncooked one. ठीक है जी. Put 5 centimeter of 3% again. So same volume, same percentage, same concentration of hydrogen peroxide, and you remember hydrogen peroxide is being used here, into a test tube labeled uncooked. So we've got two test tubes. Both of them have the same volume of 3% hydrogen peroxide. In one, we've got a fry or a chips or a potato cylinder, which is identical in shape and size, and one is a cooked potato and one is an uncooked potato. Put one of the potato cylinders into a beaker of hot water for 5 minutes. Remove the potato cylinder from the hot water and put it in the test tube labeled cooked potato. So they've also told you the measure how to cook it. So what you did is you put it in hot water for 5 minutes, and now the hot water temperature has not been fixed. We have no idea how much that temperature is. Was it 100 degrees Celsius? It just says hot water. Always think of these things when you're reading the question. So hot water 5 minutes, so no temperature has been specified. Hot means what? Then you remove the potato cylinder from the hot water and put the test tube in the one which was the cooked one. So here we put it in this one. Put the uncooked potato. So we took the uncooked one and we put it in here in the uncooked one. Now what do we do? Step 7, left the potato cylinders in a 3% hydrogen peroxide solution for 3 minutes. And then measured the height of the foam produced in each of the test tubes. So we left it there and then we waited for it. So we put it here in this also, the volume is the same, and then we waited for the foam to develop, which I'm drawing in a separate color. So uncooked will probably have some foam, maybe not has any foam, I have no idea. You've got to be started thinking of it theoretically. And then what it says, figure 1.1 shows the test tubes after 3 minutes. So this is the foam, this is the hydrogen peroxide solution, and this is the test tube, this is the potato cylinder, uncooked, cooked potato. We've got a scale in the middle, a ruler. They've given you this ruler, so look at it carefully. And then look at the diagram, and this is figure 1.1. And then it says, prepare a table to record your results. That's exactly what the syllabus says. You must know how to record a table. So measure the height of the foam in each of the test tubes in figure 1.1. Record these measurements in your table. Now you look at it, this table is for 3 marks. Now for how are you going to get these 3 marks? That is the most important thing which you've got to understand. Now the first rule of drawing a table is, okay, what are you recording? You're recording height of the foam. Now you're recording height of the foam and they could be cooked and uncooked. So how do you record it? You write the units in the heading. This units must be in the heading. You must not write centimeter here with it. This would be wrong. This would be a -1. So you do not write the units in the, so you can do it like this as well. You can do it, says a table drawn with header lines and at least two columns and headings with units and height of the foam for both cooked and uncooked recorded. Now I calculated the foam like this. I drew lines on it and I calculated it, and there's a range which is given to us in the mark schemes, which was of course would depend upon the photocopying size and how the paper was then finally made, and they would of course have that paper with them when they were checking this paper. Or you could do a table like this also. This is of course a very good way of doing it, because you see this is the independent variable. Independent means which I have chosen, and the independent variable is that you've decided to take potatoes cooked or uncooked, and this is the height of the foam is the dependent variable. So you have decided to measure the effect of the enzyme catalase, how much hydrogen peroxide is being broken down to produce oxygen, and that you're measuring in the form of the height of the foam. So that is the dependent variable. So you can do a table like this also, you could have done a table like that also, but basically what is the most important thing is that you always write the units in the heading. You do not write the units here. I'm saying this again and again. This is wrong. You cannot do this. Then it says calculate the difference in the height of the foam produced by the cooked and the uncooked potato after 3 minutes. Well, of course, you just minus it. So 3.1 minus 1 is 2.1. But then where are you going to lose marks? Well, I'm sure you are, of course, the most famous generation for making the most careless mistakes on the face of this earth. You will not write the units, and then when you don't get a mark, then you'll start screaming at yourself and the teacher and the school and everybody else except yourself. But you did not write centimeter. So if you did not write centimeter, well, then how do you expect me to give you a mark? So whatever is here, it was 3.1. Whatever you wrote here, 1.0. So you minus that, you get 2.1. So this is what you do. If you miss out the units, I'm having a lot of students when I check papers, I see a lot of students missing out the units, and that makes me very sad. Very, very sad, because that is something which is nothing but a silly, careless mistake. State one conclusion for these results. What is the conclusion that you make? Very simple. Uncooked potato produces more foam. Or you could have said catalase or the enzyme more active in the uncooked potato. So any of this would have been right. Or you said the reverse of it, that the cooked potato produces less foam, or the enzyme is less active in the cooked potato, because what has happened is they're not asking an explanation. It says, what is your conclusion? When I look at careless mistakes, what is my conclusion? My conclusion is that this student who makes these careless mistakes is not a very responsible individual. Look at his parents paying such a phenomenal amount of money for him at school, and he just goes and sits down and does careless mistakes in the paper. It saddens me. It really saddens me. Now, always remember one very important exam skill is that there's one mark. Now, you're not sure of the answer. So, if you write 2 points and one of them is correct, well, you get that one mark. So, be careful when you're writing it. You're not sure, just give me 2 points just to make sure that you get that one mark for it. Then it says identify the independent variable in this investigation. What have I changed? What have you changed? Independent what did you change? You changed the treatment of the potato. Now you decided, or I decided, that I was going to boil the one piece of potato and I was going to not boil one piece of potato. Okay, what else could I have done? I could have said, okay, let's fry one piece of potato in sunflower oil and one piece of potato in olive oil. I could have said something, okay, place one piece of potato in an acid, one piece of potato in an alkali. So here I said, okay, we're going to cook one and we're going to have the uncooked one. So the treatment of the potato is what I chose to change in this experiment. What was I going to measure? I was going to measure the height of the foam. That would be the dependent variable. But actually, what was I measuring? I was measuring the rate of enzyme activity. How much is the catalase working? Is it working more or less? So understand what is independent variable and what is dependent variable. You have to be very clear on this because there has to be a question on this in the upcoming paper. I'm 100% there's going to be a question on that. Part 2 says state why it is important that the two potato cylinders were identical in shape and size. Why would they be identical in shape and size? So that they're the same surface area. Now, I always say when you're answering this, when you answer to yourself, what if they were not the same size and the same shape? Then what would have been happened? Then of course the number of cells is different. So the mass is different. But here we said same shape and size. So then it's the surface area which is the same. Then it says part 3 state two other variables that were kept constant in this investigation. Now, what was kept constant in the investigation? Let's go back to the question. When I was reading the question, I could easily think what was the paper going to be like. So 5 cm cube here, 5 cm cube here. What is this? Volume of hydrogen peroxide was the same. Then both were 3% hydrogen peroxide. So the concentration of hydrogen peroxide solution was the same. Then we left them in it for how long? It says here you left it for 3 minutes. So the time the potato is in the hydrogen peroxide solution. So we ensured number one, the volume was the same, the volume of hydrogen peroxide solution. Number two, the concentration of the hydrogen peroxide solution. And number three, the time the potato was in the hydrogen peroxide solution. So the volume of hydrogen peroxide solution, the concentration of hydrogen peroxide solution, and the time, it was 3 minutes that the potato was in the hydrogen peroxide solution. Then part C, identify one possible source of error in the method used in this investigation. What was that? The cooked potato was not dried before adding to the hydrogen peroxide. The cooked potato was not cooled before adding to the hydrogen peroxide. The uncooked potato was not soaked in cold water. So the uneven surface of the foam, we didn't account for that. And the foam is unstable. Maybe it would increase or decrease in after how much time did we really measure it? Did we take a little while, or did we measure it exactly after 3 minutes? If we did it in 3 minutes, then the other one we did in 3.5 minutes. So think of all those points which are the sources of error in the investigation. This was only for 1 mark, but you have to come up with something sensible so that you get this 1 mark. You can't be losing this mark. Now, there was something in the question which was giving you clues about the error, like this was there in the picture, uneven surface of the foam. Then the fact that if you were actually thinking that you're doing this in the lab, the foam is unstable. The sort of bubbles burst, and after some time, maybe the surface is uneven and it decreases. But the points that they gave you, that the cooked potato was not dry before adding to the hydrogen peroxide solution, the cooked potato was not cool before adding to the hydrogen peroxide solution, and the uncooked potato was not soaked in cold water. Uneven surface of the foam. Now, what are the points that you need to remember? That you must treat them both the same way. If you heated one, then the other one, at least you heated it in hot water. It said put in hot water, so you should have put that in cold water. And then when you looked at the foam, you could see that it was not very even. And the foam wasn't going to be very stable. So the point should have come to your mind. It would only come to your mind if you think you're actually doing these in the lab. Then this is of course the one which is the most challenging for this new paper 4, which you have on the ninth D part. A student stated that catalase activity is the same in all species of plants. Plan an investigation to test this statement. And how many marks is this for? That's the most important thing you've got to understand. It's for 6 marks. And this is what I just told you in my first video on this, is that this 6 marks, everybody has to score full marks in this. You can't afford to lose any marks in this. and I don't want anybody to lose any marks. I'm going to do quite a few examples of this, so everybody has to be very prepared for it. Now, before you look at what I'm going to say, I want everybody writing down 6 points so that then you can gauge whether you've understood this or not. So the first thing you've got to understand is we're talking about an enzyme catalase, and we have to study its rate or activity. And the question is that it is the same in all species of plants. So the first thing you've got to realize is what is the independent variable? What are you going to change? What am I going to change? At least I should use two different species of plants, but you can use three, you can use four. So you talk of the independent variable. What is the independent variable? At least two different species of plant. Independent variable is at least two different species of plant. That gets you 1 mark. Then let's look at the control variables. What are the control variables? Those that have to be kept C control, C constant. Same size, same mass. Either you said same size or the same mass. Same volume of hydrogen peroxide. Same concentration of hydrogen peroxide. Same temperature, same pH. Same time. Then the tissue from the same part of the plant. Either it should from the roots or the leaves. And then the same diameter of the test tube. You realize if one test tube is thicker, now this has this much foam on it, and this has this much foam on it. Now that's not fair. The size of the test tube should be the same. The diameter or the size of the test tube should be the same. Otherwise, it's not a very fair comparison. And you put the piece of plant here, and then you put the piece of plant here. Then maybe the size of the plant piece is the same, but then the size of the test tubes is different. So there you're going to make a mistake. And then comes the dependent variable. What is going to be the dependent variable? You're going to measure the height of the foam. Then you give me another method is adding the hydrogen peroxide solution to the plant tissue. Then you prepare the plant material in such a way that the same surface area. Or if you don't want the foam, then you can collect the volume of gas using a gas syringe. And then what is going to be the control? What is going to be the control? The control is you test the hydrogen peroxide solution with no plants, or you boil the plants. And if still there is foam, then there is this your whole hypothesis is wrong, or your whole theory is wrong. Then you repeat the experiment at least 3 times so that you can calculate the mean, and you give me some relevant safety precautions. The safety precaution would be use gloves when you're handling the hydrogen peroxide. So these are some points which you can write in every practical, and I'm just going to give you those, but be careful when you're writing those. You must be not just writing those four in the beginning, you must sort of give them in the right sequence, plan an investigation to test this statement. Now, just going through the points again, once again, I have them in writing in front of you. Independent variable, you don't have to give me these headings, but you have to think under these headings. So, at least two different species of plants. Then, variables that are kept constant, same size or the mass of the plant. Then, same volume of hydrogen peroxide solution, same concentration of hydrogen peroxide, same temperature, same pH, same time, tissue from the same part, roots or leaves, and same diameter of the test tube. But look at this. Out of these, only maximum 2 marks. So there's 1 mark for this. There's 2 marks for this. So remember, there's 6 marks for this. So you're only getting 3. Even if you gave me all this stuff, I can only give you 2 marks. Then 1 mark for the independent variable. So this is 4 marks. 1, 2, 3, 4 marks. Measure of the height of the foam or measure the volume of oxygen produced. Then if you gave me a detail how to measure this, like for instance adding hydrogen peroxide to the plant tissue or achieving the same surface area, you got another mark for this. But if you didn't give me this and you gave me this, how to measure the volume of gas with the gas syringe, you got another mark. So this is, so we've got 1, 2, 3, 4, and 5, and then 1 for the control, and 1 for writing if you can repeat the experiment and you gave me some safety precaution. So how are you going to now remember this? Number one, you've got to remember, you've got to talk about the independent variable, then you've got to talk about 1 point about the control, then 1 point about the dependent variable, then some other detail if you can give it, if fine, if you don't know any detail, so you can give some other novel method. So either this or this. One, you must talk about the control. This is something which you can talk about in every practical. Then the fact that you repeat the experiment, this also you can come in every practical. And then the safety precaution is something which you can do in every practical. So you have got these headings now under you. You've got the headings under which you're going to be writing the answers. You can't give me, look at this, it says variables that are kept constant. You give me all this, it's only 2 marks I can give you for this. So you have to remember these headings under which you're going to write your answer. Your answer is to ensure that you get your 6 out of 6. There has to be a 6 marks planning question. Every child must know that. The last part of this question, part E, potatoes contain starch. Starch can be broken down into reducing sugars. Describe the test that identifies starch and reducing sugars. Everybody must have the food test on their tip of the finger. They must know them. They must know it very well. Identify starch and reducing sugar and give the results of the positive tests. So you have to give me the results of the positive test. So what were the results of the positive test? Starch test, add iodine solution, blue black. Add iodine solution and the color changes from brown. Add iodine solution, color changes from brown to blue black, right? Now for the reducing sugar, so you got 5 marks. Remember, so you've got to give me 3 points for this. So what are the 3 points? Add Benedict solution. Heat in a water bath at 80 degrees Celsius and the color changes from blue to red or green or yellow or orange or a brick red precipitate. So a very narrow marking scheme because for 5 marks there were only 5 points. 1, 2, 3, 4, 5. There's no sort of sixth or seventh point. You had to give me these 5 points to get your 5 out of 5. So for the starch is add iodine solution brown to blue black. For the reducing sugars add Benedict solution, heat in a water bath at 80 degrees Celsius and blue to brick red precipitate. Now that finishes this question and I want you to go through this video a number of times so that you can pick up all the things that I've been telling you about. I'm of course going to try and put as many videos as I can on these new topics like the planning and other things. So wishing you all the very best and thank you once again. This is going to help you in the exam skills for this paper 4.